So yeah, I'd like to welcome everyone to SINAInnovations 2016 and to the first panel symposium, or panel discussion of the symposium. And so the focus of this panel will be on therapeutic discovery and the role of medical and research centers in this process. So as you heard, my name is Paul Kenny. I'm the director of the Experimental Therapeutics Institute here at Sinai. So this is directly relevant to my day job here in terms of helping Sinai investigators translate some of the basic observations in our lab into new therapeutics. So I'd like to thank the panelists for taking the time to join us today. And as you can see from the program, really it's a stellar lineup of scientists who have expertise across the whole drug discovery enterprise from start to finish, and from an academic perspective and also from an industry perspective. And so the conversation today, the discussion today, will really be structured around a number of questions that I'll begin at least posing to the panel, hopefully to get the discussion going. And then I'll encourage everyone, I'll save some time, of course, for questions and encourage everyone to participate in the conversation. I believe, I've not used this before, but there's a QR, is that what it's called? And a QR code that will flash up on the screen. And apparently you can, there you go. You can use your phone to scan in this QR code, those of you who know how to do that. And you can ask questions electronically. They should appear on the screen, and I can work through some of the questions that you might want to ask. And those of you who don't use those QR codes, of course, there's microphones here. Those technically challenged like myself. So one thing before we get going is to notice that the questions that I try and pose, and hopefully we can get through at least some of them, may sound similar to those that you'll hear later in the day and tomorrow. And so some of the panel organizers, we got together and hopefully tried to harmonize those questions such that you'd hear recurring themes throughout the SINAInnovations symposium, really focused around areas that we thought were important. So hopefully that will help unify and harmonize, as I say, around certain themes and start a broader conversation. So before I get going and kick off the first question, I thought we'd take just a few minutes and ask the panel to briefly introduce themselves and provide just a little background on what they do. So go ahead. Yeah.
Keep it pressed. I guess I have to hold it while I'm doing it. Yeah, my name's Aled Edwards, and I head up an organization called the Structural Genomics Consortium. It's a charitable company that was founded in 2003 by the Wellcome Trust, GlaxoSmithKline, and the Canadian government. And its mission is to enable drug discovery. And how we do that is we put reagents of use to drug discovery into the public domain without restriction. It's about 300 scientists in about 6 laboratories, in 6 laboratories, University of Oxford, Toronto, Frankfurt, Karolinska, North Carolina, and in Campinas. And I think what distinguishes us is that we never file for a patent ever. Everything we do goes into the public domain without restriction. And despite that, the pharmaceutical companies that support us over the years have given us about $200 million to carry out this mission. And what we do is we solve the three-dimensional structures of proteins that are relevant for drug discovery and put them into the public domain. Together with our pharmaceutical partners, we make small molecule inhibitors of some new proteins and put that into the public domain. We also collaborate with hospitals and put these small molecule inhibitors into patient-derived cells, put all the results immediately into the public domain. And the pharmaceutical companies don't get any pre-advantage or anything. Everything is put out there. And over the years, we've had the impact, for example, the whole field of bromodomains and their druggability was really derived from our work. We've responsible for about 14% of all the human protein structures ever done on the planet. The small molecule tools we've generated are being used by 10,000 laboratories and have already appeared in 3,000 publications. And we publish, this is for the scientists guys, we publish 2.5 papers a week. And the next thing we're going to do is to try and take some of the molecules that we've invented and take it all the way to proof of concept in man as an open experiment in order to test the hypothesis that a certain target will be a therapeutic target. And so that's for the future. Thank you.
Good morning. My name's Kevin Grimes. I am co-director of a program called SPARK at Stanford. SPARK is a translational research program, and we have 2 primary missions. One is to educate our faculty, our postdoctoral fellows, and our graduate students on how to take a promising biological discovery and move it down the pathway so that we can bring it as a drug into patients. And the second mission is to actually make that happen. We take in about 12 to 15 projects per year. Criteria for entering Spark, and we ask that folks submit proposals. Number one is that it's a product that will address an unmet medical need. Number two, novel approach. And number three, which is a bit of a softer criteria, is based on how advanced the discovery is, that it's appropriate for translation, and that it has a chance of making it into either clinical trial or into an industry license within the next several years. We've now been at Spark for about 10 years or so. We've taken in about 120 projects, and we have graduated 75. For us, we consider a success either a license out or a transfer to a commercial partner or bringing something directly into clinical trial ourselves. And of the 75 graduates, we have 36 that have been transferred to the commercial sector and another dozen or so that we've brought into clinical trials ourselves. So over 60% are sort of moving in the direction that we hope. I should mention that we're indication agnostic, and we're also somewhat agnostic as to whether or not the product will be commercially attractive. About 30% of our therapeutic projects are actually repurposing previously used drugs in patients. So I look forward to the discussion, and thank you.
So my name is Ken Kaushansky. I'm currently Senior Vice President for Health Sciences at Stony Brook University and Dean of our medical school. I am a physician scientist. I am an oozing academic. I'm a hematologist by training, and several years ago I had my 15 minutes of fame in discovering thrombopoietin, the primary regulator of blood platelet and hematopoietic stem cell development. Through my career, I have mentored dozens of individuals, physician scientists like myself, and have an academic perspective on what it takes for therapeutic drug discovery and the sort of handoff between academics and industry, having worked collaboratively with a number of biotech companies and pharma in developing from the bench to the bedside and back. So I'm looking forward to this discussion and sharing some of my opinions on therapeutic drug discovery in the academic setting.
Hi, I'm Craig Lindsley. I co-direct the Vanderbilt Center for Neuroscience Drug Discovery with Jeff Kahn. As way of background, I started off in the pharmaceutical industry. I was there for about 10 years between Lilly and Merck. And then Jeff recruited me to Vanderbilt thinking that we could do drug discovery in an academic environment. And so what we've been able to build is very similar to having a small biotech nestled within university walls. It's composed of molecular pharmacology, medicinal chemists, drug metabolism scientists, behavioral pharmacology, and translational medicine scientists. So it's organized very similar to what we did at Merck. We just took out all the bureaucracy and red tape. But at the same time, we have a parallel deep science effort with our trainees, our grad students and postdocs. So for every program we have an interest in drug discovery, we may start off and have 5 to 7 years of just pure basic science around the target, developing cell lines, models, tool molecules to really understand, are these targets, are they going to have therapeutic relevance? What are the caveats and the subtleties that are going to allow us to go all the way into man with these? So our major focus is CNS, allosteric modulation of GPCRs, ion channels, and transporters. And we've been able to go all the way from high throughput screen all the way to phase 1 now in several instances, either alone or in combination with corporate partners. And so happy to talk more about that today, how that was all built.
Good morning. I'm Sutton Narula. I retired from Bristol-Myers Squibb in January, so I can truly step back and look at what's going on in the pharmaceutical industry and academia and speak my mind. I started out in molecular biology as a hardcore molecular biologist, and that was in the early 80s. And everything that could happen with the biotech revolution was going on then. And being at the Roche Institute of Molecular Biology, I got involved a lot in interferon program, which I think many of you in this audience are too young to even know of or care about. Anyway, it was the first program that I worked on, and it led to a drug. And I thought that drug discovery was very easy. I mean, I got on a program, it worked, I got a drug. It was almost 15 years after that that I actually touched a program that led to a drug. And it wasn't even homegrown, it was an in-licensed program at Schering-Plough, Remicade. I guess what I'm trying to say is, after 35 years in industry, it has always amazed me how difficult it is to get a drug out there, not just to the market, but even into a phase 3. So I'm hoping today to sort of lend my own personal views and experience that I've had over the past 35 years. 3 different companies, Schering-Plough, Hoffmann-La Roche, and Bristol-Myers Squibb, all of them very different in their culture, in the way they approach new drug discovery, and in the way they do drug development. So I'm hoping that we'll have a nice, interesting discussion today.
Good morning. My name is Rupert Vessey. I'm the President of Research and Early Development at Celgene. I'm a physician scientist by background. Celgene, for those of you who don't know, is a medium-sized biopharmaceutical company which has produced really revolutionary medicines in the fields of hematology and inflammation. My responsibility in the organization is to cover drug discovery, what we call non-clinical development, and then the early stages of clinical development. And one important thing to know about Celgene is that it's a leader in partnering activities, not only with small biotechnology companies, but also with academic institutions. And through that model, it's pioneered a whole range of very novel and innovative interactions that are distinct from what many other companies have done over the last decades in industry. So I'm also looking forward to the conversation. Thanks.
Great, thank you. So I think we'll get going. Before I start asking questions, one thing I will ask the panel, if you can keep your as concise as you possibly can. I want to squeeze in as many questions from the audience as possible. So the first question we had was, what do you think the academic medical centers have to offer in the context of drug discovery that's not already covered in depth by industry? So why exactly is it that places like Mount Sinai and other medical and research centers should engage in this type of activity? And I guess I'll throw that one out to you, I'll to kick things off. So.
Well, obviously lots of smart young people, and I guess a culture that is not staid, I would say. And I think that that's the promise of all academic centers, is the ability to think differently. The trouble is that that doesn't often come with the rigor that's required to do drug discovery. So there is a disconnect between what academics have and what's really required to make a drug.
Okay.
I think academia brings several things, as was just alluded to. Academics work in many different areas. They are not restricted to certain therapeutic arenas or certain approaches, so can work on multiple targets. And I think academia is really excellent at identifying novel targets important to disease and work in a wide spectrum of diseases. I think a second thing that particularly physician scientists can bring is a real passion to bring it across the finish line so that they can cure their patients. And I think that's really important when you're embarking on an endeavor to develop a new therapeutic. Academics often bring novel approaches. For example, the big data movement very much started in academia and is something that we can use to mine the literature and other data to identify potential new targets to go after. Academics bring on a willingness to attack non-commercially attractive diseases of the developing world. Perhaps very rare pediatric diseases. We're willing to do repurposing of drugs to bring them directly into patients when they're not going to be commercially attractive. And I guess the last thing I think academics bring is a real fearlessness, because we're not beholden to shareholders and senior management. We can take risks and really have the freedom to do that.
Great. Ken?
I'll follow up on the risk taking. One of the things that being an academics does allow an individual to do is to pursue science for science sake. And you don't know where that's going to lead. Industry is extremely good with a specific task in mind, develop this lead compound into a new safe therapeutic, and usually gets to that outcome. When you're doing science for science sake, you don't know where you're going to lead. In my particular instance, I was working on a viral oncogene, and I'm not a virologist, and yet pursuing that lead led to the discovery of a new hormone for blood cell development. The second thing that I think that academics does and allows is, in the context of the physician-scientist, the bilingual scientist, if you will, the person who brings the understanding and the passion for clinical medicine together with the skills and the rigor of a scientist to discover what is behind that particular disease and innovative approaches to it. Only by being bilingual can you, I think, translate from one arena to the other. So those are my 2 points. Freedom to pursue science wherever science takes you, and the translational aspect of being bilingual.
I think a lot of what's been said is really accurate. You know, I think the most important things that academics bring is really this passion, focus, but then time. As I mentioned in the introduction, you know, one thing we may spend 5 to 7 years with basic science, NIH dollars, really understanding a target and understanding all the caveats and nuances that allow you to develop a drug. When I left Merck, we would have maybe a year from starting a new program to delivering the first clinical candidate, and we drive that with a single assay. And you start to learn as the new targets and the new mechanisms of target modulation get more and more complex, like with signal bias. You have to have a whole suite of assays. You have to have a team of people working on those assays and validating them. And it's just something industry doesn't do. They don't have the time commitment that a basic scientist can put into it. And then the deep basic science really informs the drug discovery and enables that translation. So I think academics have a very solid place moving forward.
I would agree with everything that's been said already. I think from a personal point of view, for me, nothing was more gratifying than working with clinical researchers who actually saw patients. So while we were doing all the drudge work in the labs, trying to find medicines, to know how those medicines affected the patients was very, very important for us and really made us passionate about our drugs. But on the pragmatic side, talking about what does academia bring, especially medical institutes bring to drug discovery, I think is the treasure trove of patient samples that they might have. More and more in the last few years, my laboratories were going to patient samples much more than animal models to figure out target validation, disease pathogenesis, disease progression, and even patient stratification. So I think from a preclinical research point of view, that's a treasure trove. And I think both academia and industry should leverage it as much as possible so that we can understand our drugs better, more fully, before we bring them into humans.
Okay, so I think some really great points have been made, but let me put a little bit of a different perspective on it for you. First of all, you know, if you added up all the biologists that work in all of the biopharma companies, it's really a trivial number compared with all the biologists working in all of the academic medical centers all over the world. So, you know, what you can really discover about fundamental disease processes and selection of patients is really kind of trivial, you know, in a biopharmaceutical company. It's very difficult to do that, and you don't have access to patients, patient material, or even necessarily sometimes the disease insights that are groundbreaking. Most of the activity that goes on in discovery labs in biopharma companies, whether they're big or small, is directed to discovering and optimizing compounds against a target that you've already selected, or you're undertaking some kind of phenotypic screen and identifying a compound from that. And that's where most of the effort actually goes. So there's a huge symbiotic relationship that needs to take place between the biology and some of the discovery of those compounds. The other thing that's important to recognize is that this all plays out in fail rates. So if you look at the reasons why new compounds fail in early development, it's mostly because of efficacy. We're quite good at making drugs that have good PK properties, that can pass the safety hurdles in early development. that can be given once a day, et cetera, but it's usually efficacy that leads us to fail. And the reasons for that are, A, because we didn't understand the biology properly in the first place and picked the wrong target, and secondly, because when we get there, we choose the wrong patients to actually test the drug in. And those are two things that really can be addressed in centers like this and others around the world that can really enable this. And so partnering around those questions, I think, is incredibly important.
Okay, so I guess what I heard is that academic medical centers, like Sinai, for example, are pretty good at identifying targets. We have good basic science, and that's where you expect the targets to come from. We've got access to patient samples, which is very helpful. We got time for scientific rigor. But I guess I also heard that perhaps we don't have industrial rigor. We don't have the ability to drive things further along than, or at the same rate that you would see in industry. So for me, that I guess leads to two questions. First is, where do you really see weaknesses in centers, academic medical centers, that probably should be addressed? And where do you think that we should wisely invest to be able to fill those kind of gaps in order to help improve our ability to make drugs or facilitate this process? So where are the gaps and where should we invest? Again, I'll kick it off for you, Alan.
Well, I like to take a step back and just put out there what I think is that we have a system for the past 30 years that have been investing more and more to drug discovery, and the number of new medicines coming out is flatlining. This is the reason that industry is outsourcing its research. At a fundamental question, you have to ask yourself, is there any reason to believe that if that activity is offloaded to academic institutions that it's going to be economically as viable? It's a fairly good move by industry to offload its risky stuff to academia, but that means we're going to be spending a lot of money to fail a lot. And so every academic institution has to go in eyes wide open. This is a business about failure and spending money and not making a success. And this is, we do have all the assets, like the patient cells, for example, which is an interesting thing, but if we treat them as assets, as we also do, that's kind of odious, right? Selling human flesh as an asset to be partnered with industry. I sort of think, so what do I think we should do? We should just focus on the biology and not honestly try to do too much in drug discovery, because as Rupert just said, the big need in drug discovery is more biology, right? And if we start to mix up learning more biology with trying to create a sustainable academic institution, I think we're going to get into trouble.
Kevin?
Sure. We have a lot of weaknesses in this enterprise as academics. One is incentives. We're incented to publish. We're incented to get promoted. And developing a new therapeutic doesn't necessarily fall into that paradigm, and I think maybe that's something that needs to be revisited. Number two, we lack funding for applied science. The grants tend to focus much more on discovery science and occasionally on later stage clinical trials, but money for applied science is not there. Furthermore, we're not trained in the applied science in academics, and that's something that we can remedy through education. And another thing that I would bring up is just the way that we think. Academics like to take the random walk and follow the science where it's most interesting, and I think that's fantastic for discovery science. But once you've set your mind on developing a drug, you have to start with the end in mind. You have to follow more of a project management mode. You have to be much more disciplined. You have to develop your target product profile right up front. That's going to include all of those features that you need to bring it across the finish line. So I think those are very important. I could go on, but I'll turn it on to the rest.
So my gripe is with deans and academics who believe that the rugged individualist is the only way to succeed in academic medicine. One of the things we lack in academics that I think would move our processes forward, and I do agree that limited is too strong of a word, but should focus more on the discovery, the target identification, and not so much the drug development per se. But I think our major weakness is, in order to be tenured, you have to be the first or senior author on every paper. You have to get 1 or 2 or 3 R01s, individual research awards. We do not value team science in the academic process, and we ought to be taking a lesson from pharma, from startup biotechs, where team science is the watchword, where team science does succeed in getting to discovery much more quickly. And so I have, wherever I have been, a department chair or a dean, have really focused that team science is important, and that being a vital member of a team that is contributing something that no one else can contribute is just as good as the first authored paper. I think if we adopt team science in our efforts towards therapeutic development, target identification, we will be more successful than force functioning people into silos that might not be as productive.
Well, I definitely agree with that. Big team science is critical. It's important. All the different disciplines have to work together seamlessly to make these things work. And so obviously in the academic environment, classically, that's a huge problem, as was just outlined. Another big thing we face is that functional groups like DMPK that are really critical to drug discovery are very poorly funded by NIH. General medicine has very few funding opportunities for DMPK scientists. And you have to have those in your team, but you have to figure out alternate ways to fund them because they can't get really high levels of R01 funding or other kind of funding. And again, without DMPK, you really can't develop a drug. And I think the third big issue where we have a weakness is across, I think, every academic medical center is your technology transfer office, just in terms of their willingness to patent, their willingness to nationalize cases before you've partnered, or feeling pressure, getting a lot of pressure to partner early because of nationalizations coming up. And then just the negotiation time. A lot of university transfer offices are notorious among big pharma as being really challenges to work with and overvaluing their assets and things of that nature.
Yes, so just listening to the previous speakers, what becomes apparent to me is that we all speak the language of science, but we are living in different worlds. What the pharma world is goaded by, the objectives and agendas and goals, are very different from what is true for academicians, especially in view of getting grants and papers published, etc. So I really don't know what the solution to that is, but I do believe that you don't fix what's not broken. So if pharma has the ability to do team science, maybe that's what pharma should do. And really, I don't think in 35 years, I've literally changed fields, and it has been impossible for me to gather a team of experts overnight to take on a new area. And for that, you have to go back to academia and find a group that has the expertise in it. So I believe that somehow we have to find that middle ground where academia and industry can do what they're good at. And talking about patents and, you were talking about patents and nationalization phases, et cetera. One of the biggest problems I've had in my collaborations is research agreements. It takes months, sometimes a year, to put together a research agreement. I think the academia should look at what they're doing vis-a-vis legal agreements and see whether they're making them so onerous that all it does is make the lawyers go back and forth, back and forth. That has been the biggest deterrent to us picking up a program quickly and making it move expeditiously. So I think that's an area, both industry, I think pharma is equally bad, both industry and academia should take a look at.
Well, I guess if you're running an academic institution, which I've never had to do, but while we're talking about it, it's making me think about it, I would imagine you have to take a step back and think about what it is you're actually trying to accomplish by engaging in an endeavor of this type. And, I would assume that, or at least it doesn't make much sense to me that you would want to rebuild all of the functions that are actually incredibly well done in the biopharma industry or by CROs, including, non-clinical development, late development, large-scale medicinal chemistry. It wouldn't make any sense at all to try to replicate any of those things. And then I think you have to say, well, if we're going to participate in this activity, what kind of reward would I like to get back from the contribution that I'm making? And I think if you can lay that out at a strategic level in your institution, then it can drive a whole lot of things downstream, including how your tech transfer office should behave, what kind of agreements you want to strike, what you want to see in those agreements so you can have a whole strategy. And then the same thing applies to how you line up your biology, your translational thinking. And one other observation that I would make is that in my time in industry, which is now getting on for 20 years, it used to really be just biology, but of course many academic institutions have fantastic structural biology departments. They have actually very good chemists. So there are some here who've joined academic institutions who've been trained in some of the best companies in the world. And I've really noticed actually that at least the initial insights into how one might drug a target are becoming more and more innovative within the academic institutions that we work with. So that's definitely a sort of sweet spot that takes you one step beyond the biology in terms of creating those initial compounds that will allow you to ask the right question and provide real starting points for a drug discovery program. So I think you really have to think, you know, what is it you're trying to accomplish in your institution, and then design the systems that you have to play in the places that you want to play, and then make sure that you participate in the downstream rewards of that activity in the right way.
So we've heard a little bit about this in terms of some issues that you may run into with tech transfer offices, but I was curious to hear other personal experiences you may have had either on the industry side or an academic side in terms of challenges that you may face, your own personal challenges that you've seen when trying to develop a drug, or you've seen a drug being developed in an academic setting, and also opportunities, areas where you think that this is something that perhaps should have turned into a drug, should have been translated. So do you have personal experiences that could speak to the positive and negative beyond tech transfer offices in an academic setting? Again, I'll ask you that.
So I think that, let me share with you an example from McGill University. There's a neurology hospital there called, it's a famous one, the Montreal Neurological Institute. And what it's decided in order to participate better in the development of new medicines is for the next 6 years, they've agreed, as an experiment, not to file patents on any discovery internally. And so they're not going to have any MTAs, not going to have any ownership over intellectual property, and they're going to just share their science with everybody. They have incredible patient cohorts. They see very large clinics in ALS, in Huntington's, MS, Parkinson's. And the idea is that if we treat the science and the patient resource as a common good, then it's to industry's advantage to come work with it, as we know, because the big problem is sorting out the biology. So that's kind of a cool experiment, and we'll see how it happens. But, I mean, they're in essence, trying to do what they do best and let industry does what it do best, and remove the barriers which were the tech transfer offices by eliminating them.
Okay.
I sort of want to mention, I think how well a tech transfer office works depends on how it's motivated.
I should say, tech transfer office here at Sinai is outstanding. If Eric leaves the audience, a fantastic tech transfer office.
And
we have no complaints.
I truly find our tech transfer office at Stanford mainly motivated at transferring discoveries to benefit society. And, you know, in terms of financial upside and so forth, they have a reputation for being extremely easy for establishing industry partnerships and licenses. So it doesn't have to be hard. Sometimes not patenting something will ensure that it's never developed. And you have to keep that in mind. There's a real balance between when you patent and when you do not. And I've seen fantastic programs when I worked in the industry side that couldn't move forward because there was no way for someone to capture any return on the very expensive investment it would have taken to bring it forward as a drug. So I think that's really important to understand. We've been reasonably successful at Stanford, and I think the opportunity that we've taken advantage of is really the generosity of our industry colleagues. So Spark is a program that's really embedded within the medical school, within the university. And we have invited folks from the local venture community, folks from the local biotech industry, folks from local pharma, who come in every week. We have over 100 volunteer advisors who have all signed CDAs, who all sign over whatever they may invent. We ask them not to invent to the university. And they work with our academic teams. And we have this combination of extremely great basic research. You heard an example of what Carl was presenting. And then really seasoned veterans who know how to take this and turn it into something that may benefit patients. So at some level, the dichotomy between academia and industry doesn't have to be there. and there are definitely ways to overcome it. And the success of Spark is, 90% industry volunteers who are motivated primarily by altruism.
So I'm going to use this opportunity to vent one of my pet peeves that I have seen over my career in academics, and that is the singular focus of junior academic individuals to focus on the patent and the Office of Technology Transfer in exclusion to the major mission, what I believe the major mission to be academics, which is the open discourse of discovery. We are mostly funded by the National Institutes of Health, which means taxpayers. We do have a responsibility to share what we discover. And now I'm going to be speaking mostly to the students who in the audience who are interested in careers in academics, to the junior faculty members. I have seen way too many individuals who find something in the laboratory, believe they're going to become a billionaire because of it, and are busy writing patents and not papers. Please avoid that. The likelihood that you are going to become a billionaire based on your discovery in the laboratory is low. I promise you. Work with the Office of Technology Transfer, whether it's good, bad, or indifferent, but keep your mind on the focus of new discovery. There is nothing more heady than being the only person on the planet who knows your discovery, and then sharing it with others. The rewards are immense in the purer pursuit. So don't get trapped writing patents and not get promoted. Don't get trapped writing patents and not getting tenure. There's a lot of rewards in academics in discovering. In my particular case, I did not gain a single cent from my major discovery, and yet when the drug was licensed by the FDA for use in patients, I felt an overwhelming sense of satisfaction. So focus on, in academics, focus on what is the primary mission, which is sharing our knowledge with others. Don't just focus on the patent. So that's my pet peeve, and I'll get off the soapbox now.
So a really positive thing. So we've had instances now with 4 programs where we were directly competing with Big Pharma on the same target. Big Pharma ended up dropping out. And because we had such a long-standing deep basic science effort, we understood caveats to the target, the way to modulate the target, and signal bias. And we were able to take compounds all the way through IND-enabling studies and into the clinic, where Big Pharma failed. Because again, the deep basic science that we bring is why it's so important to have the chemistry, the DMPK, and the pharmacology in an academic setting to push molecules that really engage targets differently and can be successful. I think one of my big pet peeves is when you're in a soft money medical school and you have to pay for everything. So Jeff and I, there's been no endowment, we pay for everything. You really have to balance your federal funding, your foundation funding, and then sponsored research with companies. And you're always, it's always a risk to partner too early with a company and have them sponsor research because things change. We've had numerous companies now, and this is, in the last 5 years, have very suddenly exited neuroscience, for example. And there's no warning. You get, everything's going great. Then the next day you get a call saying, we got a new CEO or a new head of science. We've exited neuroscience. We're going to return your program. Which is why it's so important when you do contracting with Big Pharma from a university setting, you have to have really strong diligence terms so that if something does happen, all the IP comes back, all the rights come back to you. But even when that happens, your project just gets really delayed. I mean, you anytime that happens, you lose 6 to 9 months of productivity while you're waiting for all the contractual stuff to occur. So when you do make deals, having strong diligence terms on return of everything and having rights to operator is critically important.
So as far as patents are concerned, I have many to my name, and I would say maybe a couple are of some value. So I think it's a great point. You can file patents, but they're only as good as what is in them. One of the things that I do want to mention is that I think we've become too scared to share our science with each other sometimes in early phases, and that really does impede progress. When I was in Schering-Plough, it was the sort of the burgeoning days of cytokines. And what we did was we provided everyone who asked for it, most of the people who asked for it under a very simple MTA, IL-4, GM-CSF, IL-10, etc. IL-4 and GM-CSF were used by hundreds of investigators, and it really brought about a lot of progress in the dendritic cell biology. IL-10, which is still a favorite cytokine of mine, never made it into the market. I mean, we would never have learned all we know about this intriguing cytokine, which is still of so much interest to people, if we hadn't given it out to investigators. I think we need to do more of that on both sides. Sometimes it saddens me when I see people in academics becoming so, closed up about their science. So I would agree. I don't think about filing patents, think about publishing your data. I've had very, very positive experience working with academia, but usually when I'm working one-on-one with an investigator. It's always been very fruitful. We've learned something from it. We've moved on. I think it is the larger collaborations where several different laboratories from a university might be involved that it becomes hairy. And that's where I think the differing objectives and agendas and goals start clashing with each other. So I think the only way to manage that, if academia is interested in doing those collaborations and if industry is interested in making use of it, is to impose some sort of project management kind of stuff that we use in industry. It is helpful. It's not always very successful, but it's helpful in keeping a program on its path. So I think those kinds of measures might help if that's what the academic's goal is.
So one of the experiences that I've had, and probably everybody else on the panel has had, is the challenge of reproducing initial research findings that look incredibly promising and might lead to a new therapeutic avenue. And now there are several publications that demonstrate that, about half the time you actually can't reproduce the result, and that may be because of reagent variation or other reasons that are not entirely clear. So one of the things that I think is helpful in this space, particularly around these early observations, obviously is replication, but also is the provision of some funding and in particular tools and skills that enable you to have exactly the right things to demonstrate that that initial result is incredibly robust and therefore really worth investing in. Because once you make that decision, there's a lot of effort downstream that starts to be incurred. So that happens. It happens, reasonably often when we take things in and try to reproduce them. And we've tried to address that through the provision of a number of incubator-type funds that fund a different type of activity from what you would get from an academic funding, but allow you to have access to create tools and then really robustly reproduce a promising result and start whatever you do downstream from there on a firm foundation.
Okay, I'd like to open the panel up to any questions that the audience may have. So we have one over here.
Hello. Can you hear me? Okay, great. Hi, everyone. Yeah, thank you for that discussion. I just had a generally pretty broad question. What is the most important drug that has not yet been developed, and what will it take to develop it?
I'm not answering that question. That's a tough one. So anyone would like to take that one on? Oh, wow, we have a lot.
Say for a given area, so one of the reasons we focus on neuroscience is all the standard of care drugs that are the most effective medications in Parkinson's, schizophrenia, were developed in the 50s and 60s, and they're not very well tolerated. There's lots of rooms to improve, and mental health in general is in shambles, and there's lots of needs for fundamentally new medications in those spaces, but by no means is that the end-all, be-all. That's just my more CNS-centric focus, but it is a major unmet medical need.
Dennis?
I think we're going to talk about that actually in one of the panels.
Yeah.
Drug discovery in neuroscience. So, in case anybody from Mount Sinai was listening to the panel, it's okay to patent. We want you to patent. And you can publish too. There's a way to publish, patent, then publish, patent, and then publish. Now, I made a mistake in my career at one point where I published and then patented, but we want you to protect the great science that you're doing at Mount Sinai, but you can also publish and get tenure and so forth. And in fact, for those who don't know it, I've instructed the Appointments and Promotions Committee, over the years to broaden, what it takes to get promoted. And it's not simply, having first or last authorship. We've emphasized team science so that you get credit for being part of a team and publishing together, and you get credit, for patents and intellectual, other forms of intellectual property. Because as I mentioned in my opening remarks, we're all about doing great science at Mount Sinai, but ultimately, in partnership, in many cases, getting it to the patients who need it most. So just want to clarify for all the Mount Sinai students. My question is, and you kind of addressed it a little bit, is, and we've struggled with this, what is the right investment that an institution like Mount Sinai or Stanford, and others, should make to synergize with industry and ultimately together do a better job of getting drugs to the market, because it is hard, and that's been mentioned. Some of the ideas that came up that maybe you want to elaborate on, should we invest more in biorepositories so that we have more patient-type samples to interrogate with our own science, but then in the right way, in an ethical way, to share, with industry? Should we invest more in big data analytics? We have one of the largest health systems in the country, if not the world here, and using big data analytics to look at the enormous amounts of clinical and research data. Will that further enhance, therapeutic discoveries? We don't want to replicate what pharma does great, which is chemistry. We're not going to hire an army of chemists. So, but what is the right footprint for chemistry and monoclonal antibodies and so forth? So I'd be interested in your opinions on that question. Thank you.
So on your questions about repositories and big data and analytics, I mean, those things are great and they're really useful provided they come up with actionable answers. So they're not useful investments just to sort of have them, and there are places in the world that actually do that kind of thing. They just sort of collect everything up and then they come along and say, well, what should we do with that? What we really need, in my opinion, and what works incredibly well is when you can look at a disease or a condition and you can use those kinds of things that you described as investments to segment that population at a molecular level. So when we see something like that, we see a complicated disease being classified by pathway or some kind of molecular angle, that opens up a huge opportunity for drug discovery, because that's really how we discover drugs. And so then you've got an avenue in where you can actually action something, and most important of all, you've got a question that you absolutely know that you're going to ask when you get to the clinic. Because the thing that I hate most is just pushing molecules into the clinic and having teams turn around and tell me that, well, we'll see a signal and we'll know it when we see it, because we never do. And it just goes on and on and on and wastes an enormous amount of money, and people don't like stopping the program. So you have the substrate in terms of talent, patients, access to those things. I know here the genetics and informatics is absolutely spectacular. And if you can bring those things to bear and provide those kinds of insights, those are then really actionable from a drug discovery and early development point of view, and that will increase our success rate.
Okay. Steve?
So I just wanted to turn things around a little, and just from the perspective of cancer. Yes, we do need new drugs, but we have lots of drugs now. We have drugs in immunology, targeted therapies, radiation, chemotherapy, and we do a terrible job in how they're used, because this is going to be a combination. We know from cancer, it's multimodality therapy. The problem part is very hard to develop preclinical screens. This disincentivized, because if you have 3 or 4 drugs that would work phenomenally, there's going to be 3 or 4 companies that have to get together to do that. So we're tending to do right now, we're at the next phase of 1+1. And if you look in the literature, anti-PD-1 plus X, you see hundreds of trials being done there. That's pretty simplistic at this point. So the question, is there going to be methods of looking at the impact or the effect biochemically and various ways to say we're going to be able to do screening with 3 or 4 drugs, think about the number of iterations already of the existing drugs, and find a way both economically, because it's going to take companies getting together with academics, to come up with the right combination that's going to give us the best results with the existing drugs at this time.
Steve, I think there are a lot of efforts to try to do that. So I mean, everybody recognizes what you're saying and recognizes that the way that combination therapy is done today is not optimal, and not only that, but the way that early development is often done in oncologic disease is not optimal either, because you have to sort of start with a basket of all-comer patients who failed multiple therapies, and I don't know how many of the things that have been brought into development and have failed because that was the patient population where you had to start, but that wasn't the idea that the drug was originally conceived of. So I think there are, I don't know all of them, but I do think there are quite a lot of efforts to, and innovative trial designs to try to address your question. And I think that this is another area where, perhaps more sophisticated preclinical work and potentially informatics-based approaches might actually direct us to things that are more fruitful. But in the meantime, there are other hurdles like regulatory hurdles and all kinds of other things that sort of impede doing this in the way that we would really like to do it. But it's not lost on people that this needs to be addressed.
I'd really like to take one using this QR scanner, my first time doing this. So I'm going to, one from the audience, so before we move on with a question. So maybe Craig could take this one. So in which ways do you incentivize big science, team science, I should say, in an academic setting? Because that's not traditionally how academia has been structured, to reward people who work as part of a team. And I know that's something you spent a lot of time thinking about.
So we really struggle with that, and we had to talk to a lot of chairs and deans and really try to explain how this is going to work. And then our center, while we have, it's comprised of multiple faculty, we all have a single mission. And we've really managed to get our institution to really value the big team science for promotion, co-author publications, multi-PI grants, co-PI grants. It wasn't easy because it really is a very large culture change, but by doing that, people are getting tenure, people are advancing, and it's working out really, really well. And then it really, just like in a big pharma, everyone is focused on a singular goal, and everyone participates and delivers to achieve that goal. It's not always easy to do, but if you're going to do this in a university setting, you really have to spend quite a bit of time on that aspect, or it's not going to work.
Okay.
Can I just say one thing about the chemistry that got brought up? For anything you're going to do in the drug discovery space or the chemical biology space or target validation space, chemistry is really important. Notwithstanding patented molecules that companies would want to license for the composition of matter, which is so important, but also just having medicinal chemists that partner with biologists making tool molecules. And that's what was so important for us, is having early on tool molecules across multiple chemotypes with multiple modes of pharmacology to really go in depth and study the program, study the target, study the biology of the target. That's all great fodder for federally funded grants. And then if you even start to think then, that'll help you decide if you want to go and develop to have a drug discovery program. And is it appropriate for a small molecule or a biologic? And if it is small molecule, if you don't have chemists, and you can't really take advantage of biomarker discovery, making radioligands, PET tracers, spec agents, all that kind of stuff. And so, I don't think you need to hire 100 medicinal chemists, but you need to have a medicinal chemistry team in place that can really partner with biologists and advance their programs. Yeah. There's really no other way to do it.
Yep. Yes, go ahead.
And just for the Sinai people, you have Jian Jin here, is a professor here, a med chemist, and he's one of the best on the planet. So if you guys ever want to collaborate with anybody, go over to him, and he gives away his chemistry to the world without restriction.
Okay, so we're almost out of time.
Hi. So we are in the election season and criticizing a lot of things which federal government should do or should not do. But one of the things which has come out recently from the federal government, which can be very focused towards team science, and this is building teams across the industry and the academics, which is the NNMI initiatives, which is the National Notes of Manufacturing Innovation. And there is one specifically in biomanufacturing, which is very geared towards pharma. And here the money only becomes available if there is a public-private partnership. So if you come and bring a third partner in this equation, industry, academia, and then federal government or any other agency, which can give a resource up. The resource is only available if there is a partnership. And it breaks down the barrier of IP as well, because the IP in this NNMI is regulated centrally, and all the partners have to agree to that. So I think there are some interesting modes which are coming very much in line of team science, which may help us to address this problem and think of team as a much bigger entity than a smaller localized one.
Yeah, and rather than, so we can get, I know we're out of time, just a comment from me at least. I think that's an innovative way of supporting this type of academic endeavor where you have these public-private partnerships and you bring people together as a team where there's investment from all partners and then IP is handled in an appropriate manner. I agree completely. So I know we're out of time. I just wanted to take one more question if we could, because I know you've been there for a while.
Thank you. So this is kind of going back to the last question you had about being scared to share data or share science. Earlier this year, the primates lab group at the Primate Center at the University of Wisconsin, they started sharing their data online for some Zika trials that they're doing. So kind of going back, so kind of relating to these past few years where you've had a lot of time-sensitive need for these therapeutics, do you see this more openness in data, helping with these production of these therapeutics? Because I don't really see it happening in more chronic diseases like cancer, but with a lot of these infectious diseases where these things are very time-sensitive with Zika and Ebola. Do you see that becoming more, I guess, systematic, or do you even think that's a good idea?
Do you want to take that? Perhaps you could use your...
We've been talking about the paucity of new drugs, right? I think pharma has thrown billions of dollars into research. If you just look at the research budgets of pharma, it's mind-boggling, and so is academia. I think that the complexity of diseases that we are dealing with, especially the chronic diseases, probably throwing everything at ed that academia has and industry has is going to be required for us to get that next good break in medicine. It could be combination therapies. I mean, the point made earlier about oncologic drugs, where immunotherapy, PD-1, Yervoy, et cetera, have made such a breakthrough. But going forward into combinations, there really isn't much science guiding it. A lot of it is being done empirically because I think we're not working together. So, I hope that that's what happens, because I think just doing it alone is not going to get us there. I think just having dealt with chronic diseases like rheumatoid arthritis, multiple sclerosis, and now fibrotic diseases, which was the last area I was working for in BMS, these are such complex diseases that single pathway drugs are probably not going to do it. So I would hope that we move more and more towards that kind of teamwork between industry and academia.
So unfortunately, we're out of time. Please join me in thanking the panel for a great discussion.